Hi everyone, my name is Sujin and I'm a PhD student at Ajou University. Today I'm going to talk about my research, and the title of my research is Exploring the Interplay Between Metabolic Syndrome and Brain Volume in Depression. Multimodal research has been a key component in medical studies. By integrating data like brain images, clinical records, clinical notes, we can gain a more complete picture for better health research. Alongside this, multi-organ interaction is gaining attention because our organs don't work in isolation. Understanding how the brain, heart, and gut connect give us insights into disease and health that single organ studies might miss. So OHDSI2024 is especially useful in this context. Its standardized format makes integrating diverse types of data more efficient. So there were successful reconstruction cases like radiology CDM and genomic CDM, enhancing the potential for large-scale multimodal research. However, there are still limited use cases, particularly those involving multi-organ interactions. Despite limited research, our research team has been actively making great efforts to bridge this gap. This year, we published two papers demonstrating the power of multimodality by developing prediction models for treatment-resistant depression and suicidal thoughts using medical records and clinical notes and brain images. The study that I'm going to present today is also a part of our recent efforts, and this shows how we used OHDSI2024 to explore brain-body interactions in depression and metabolic syndrome. I developed classification models for metabolic syndrome in patients with depression. Depression and metabolic syndrome are closely linked, sharing mechanisms like inflammation and neuroendocrine dysregulation. This suggests a bidirectional relationship where each condition can worsen the other. So despite this, the potential pathophysiological connections between metabolic syndrome and depression haven't been extensively explored. So building on previous findings that the brain may act as a mediator, we investigated brain-body interactions to further understand this connection. So we started this study with these two hypotheses. First, that interactions between brain volume and blood lab variables could explain the link between depression and metabolic syndrome. And second, that these interactions could identify depression subtypes, helping predict their risk for metabolic syndrome. In this study, we followed these three key steps. First, we reduced brain regions using non-negative matrix vectorization, which is called NMF. And then we used canonical correlation analysis to examine the interaction between brain and body using both brain image features and clinical features. Finally, we applied clustering analysis to identify patient subtypes of depression and developed classification models. Using the OHDSI2024 database of Ajou University Hospital, we comprised a study cohort of patients with depression. We excluded patients without brain MRI images, and we excluded patients with those who had previous psychiatric diseases. So after pre-processing, the final sample consisted of two groups, 76 with metabolic syndrome and 74 without metabolic syndrome, respectively. And NMF, which was used to reveal, key networks within the brain, is a method that determines how brain networks are organized. For the CCA, we use 6 metabolic syndrome variables and 38 blood test features to analyze how these interact with the brain networks identified from NMF. This allowed us to reveal important brain-body interactions that contribute to metabolic syndrome classification. Then, to identify potential subtypes of patients with depression, we used K-means clustering based on the first CCA component. We determined the optimal K by using metrics like Silhouette score, and this resulted in 4 depression subtypes. For classification, we used XGBoost, and 3 models were developed. Model 1 with demographic features and metabolic syndromes, and Model 2 adding more VVM NMF components, and Model 3 incorporating subtype. So through NMF, brain volume was reduced to 4 components, corresponding to previously known structural brain networks. And through CCA, 4 distinct components were identified with loadings corresponding to clinical variables and structural brain networks, which equal to 4 NMF components, respectively. And Model 1 had an AUROC of 68, and while adding NMF components in Model 2, improved it to 72. And Model 3, which included subtypes, performed the best with an AUROC 75. So across all metrics, Model 3 showed the highest performance. So this is research summary. Our initial exploration of brain-body interactions using CDM was instrumental in understanding the impact of this multi-organ interaction on health. We found that these interactions can be effective classifiers, and we believe there's much potential to apply this approach to other domains and other samples, offering deeper insights into how multi-organ interactions influence health. So thanks for listening, and I'll be really happy to discuss the clinical interpretations of these found brain networks and their roles in the subtypes during the poster session. Thank you.